Let's look at the steps of DNA replication in more detail. To begin the process of DNA replication, the two double helix strands are unwound and separated from each other by the helicase enzyme. The point where the DNA is separated into single strands and where new DNA will be synthesized is known as the replication fork. Single-strand binding proteins, or SSBs, quickly coat the newly exposed single strands. SSBs maintain the separated strands during DNA replication. Without the SSBs, the complementary DNA strands could easily snap back together. SSBs bind loosely to the DNA and are displaced when the polymerase enzymes begin synthesizing the new DNA strands. Now that they are separated, the two single DNA strands can act as templates for the production of two new complementary DNA strands. Remember that the double helix consists of two antiparallel DNA strands with complementary 5' to 3' strands running in opposite directions. Polymerase enzymes can synthesize nucleic acid strands only in the 5' to 3' direction, hooking the 5' phosphate group of an incoming nucleotide onto the 3' hydroxyl group at the end of the growing nucleic acid chain. Because the chain grows by extension off the 3' hydroxyl group, strand synthesis is said to proceed in a 5' to 3' direction. Even when the strands are separated, however, DNA polymerase cannot simply begin copying the DNA. DNA polymerase can only extend a nucleic acid chain, but cannot start one from scratch. To give the DNA polymerase a place to start, an RNA polymerase called primase first copies a short stretch of the DNA strand. This creates a complementary RNA segment up to 60 nucleotides long that is called a primer. Now DNA polymerase can copy the DNA strand. The DNA polymerase starts at the 3' end of the RNA primer and, using the original DNA strand as a guide, begins to synthesize a new complementary DNA strand. Two polymerase enzymes are required, one for each parental DNA strand. Due to the antiparallel nature of the DNA strands, however, the polymerase enzymes on the two strands start to move in opposite directions. One polymerase can remain on its DNA template and copy the DNA in one continuous strand. However, the other polymerase can only copy a short stretch of DNA before it runs into the primer of the previously sequenced fragment. It is therefore forced to repeatedly release the DNA strand and slide further upstream to begin extension from another RNA primer. The sliding clamp helps hold this DNA polymerase onto the DNA as the DNA moves through the replication machinery. The sliding clamp makes the polymerase processive. The continuously synthesized strand is known as the leading strand, while the strand that is synthesized in short pieces is known as the lagging strand. The short stretches of DNA that make up the lagging strand are known as Okazaki fragments. Before the lagging strand DNA exits the replication factory, its RNA primers must be removed and the Okazaki fragments must be joined together to create a continuous DNA strand. The first step is the removal of the RNA primer. RNase H, which recognizes RNA-DNA hybrid helices, degrades the RNA by hydrolyzing its phosphodiester bonds. Next, the sequence gap created by RNase H is then filled in by DNA polymerase, which extends the 3' end of the neighboring Okazaki fragment. Finally, the Okazaki fragments are joined together by DNA ligase, which hooks together the 3' end of one fragment to the 5' phosphate group of the neighboring fragment in an ATP or NAD dependent reaction.